Before translation begins, a 5' cap of G3P and a 3' cap of adenine nucleotides are added to the RNA. The ribosome is made up of a large and small subunit. Inside the ribosome are 3 sites. The first site is called the A site, and this is where tRNA is accepted into the ribosome. The second site is called the P site, and this is where the polypeptide chain is built by adding amino acids. The third site is called the E site, and this is where the tRNA exits the ribosome. To begin translation, we will begin with the codon AUG, which is the start codon and also codes for methionine. tRNA carrying the anticodon for AUG enters the A site, carrying with it the amino acid methionine. As we move on to the next codon, this tRNA moves to the P site. We now here have the codon CCA. The tRNA with the anticodon for CCA will enter the A site, carrying proline. As we move on to the next set of codon, to the next codon, proline will begin to move towards the P site, where methionine will attach to proline, and this tRNA will move to the E site, where it will eventually exit. This codon, UGG, will receive tRNA with the anticodon ACC carrying tryptophan. As we move on to the next codon, this tRNA will move to the P site, where these 2 previous amino acids will add on to this tRNA. This tRNA will exit, and this tRNA will remain in the E site. We now have AAG. The tRNA contained with the anticodon UCC will enter the A site with the amino acid lysine. As we move on to the next codon, this tRNA will move to the P site, where these amino acids will add on to this tRNA. This tRNA will exit the ribosome, and this tRNA will move to the E site, waiting to exit. Our last codon is CUC, where we'll receive tRNA with anticodon GAC carrying the amino acid leucine. As we move here, as we move along, we move to a stop codon, UAA, which means no more tRNAs will enter the ribosome. Leucine is the last to move to the P site. Acids will add on to it. All these tRNAs will exit. The final result is one polypeptide made up of 5 amino acids.